few minutes after the hour. So hello everyone and happy Monday. It's my honor to introduce today's speaker, Katie Davis, Dr. Katie Davis. And this seminar is co-sponsored by the Cancer Institute and the Cancer Institute DNA Damage and Host Response Program. And as you may know, a component of this program studies the effects of cancer therapy on the host's normal tissues, so the non-cancer tissues, in addition to studying tumor effects. And Katie's research falls right in line with the research that is being done here on campus. So I'm excited that she's giving this talk to us today. She received her Ph.D. in behavior, cognition, and neuroscience from the American University in Washington, D.C. And after that became a postdoctoral fellow at Johns Hopkins. And in 2014, she established her own lab at Johns Hopkins. And her lab has been funded continuously since then, uninterrupted from various research sources. Her research has a focus on ionizing radiation, brain injury, and cognition. And what's interesting is that she applies those radiation biology concepts to different scenarios of radiation exposure. So that's not just radiation therapy, but she also has several projects in the lab where she studies the effects of ionizing radiation on astronauts as they travel in deep space. And that work in astronauts is acknowledged nationwide, what she does. And in 2019, she was awarded a merit award from Brookhaven National Laboratory, and that is a lab that does a lot of radiation research for NASA for what they said indispensable contributions to the long-term health of astronauts. So that was a very highly rated award that she received. Just last year, you can see her on the screen, she became a faculty member at Uniformed Services University of the Health Sciences and AFRI. And these are institutions that have a very long history and a core of faculty who all study normal tissue radiation responses. So she found a good home there. And she also became adjunct professor at the University of Maryland Global Campus in Adelphia, Maryland. So with that, I would like to ask Katie to give her presentation and looking forward to hearing about this research today, Katie.
Great. Thank you very much. Today I'm going to talk to you about a study we did looking at tumor responses and normal tissue effects in a mouse model. So like Marianne said, I'm primarily interested in cognitive deficits following radiation exposure. But first, these are my disclosures. These are my opinions and assertions, and all the animal work here was performed at Johns Hopkins, and it was all approved by the JHU Animal Care and Use Committee. So when we think about brain damage from radiation-induced injury, this has evolved over time. And decades ago, the brain was viewed as relatively radio-resistant, with large doses being required, as you see here, to cause damage. And that damage was primarily viewed as things like necrosis and breakdown of the vasculature. So those early studies were attributing these effects to reduction in the proliferative capacity of glia and vascular cells, and that these were progressive and irreversible, the changes that we were seeing. As we moved on to fractionated partial or fractionated whole brain irradiation, we still see that patients report clinically significant decline in their cognitive function long-term, so 1 to 6 months. And for many patients, this remains after their therapy is over and concludes. So what we see with these patients, though, is that there isn't always a decline in some of these other measures that we see with very high doses of radiation. So that makes us wonder, what is the mechanism of these deficits? These things are important because patients are now surviving longer due to improved therapies. And this population of patients with cognitive side effects is growing. And it is now recognized as second only. Their quality of life is recognized second only to their survival. Oops, sorry. So this is sort of the timeline of radiation-induced cognitive impairment. these acute effects we rarely see now because of fractionated radiotherapy and what I'm going to talk today about stereotaxic radiosurgery. So we rarely see these effects, but we still see these early delayed and late delayed effects. And these late delayed are primarily the ones that are a problem because the cognitive deficits seem to persist for more than a year. And for many, they're progressive and irreversible decline. So many of these things in the early stages can be treated with steroids. So we believe they're inflammation dependent. And here, when we get to the late delayed effects, you see things like vasculopathy, demyelination, loss of oligodendrocytes and neuronal precursors, gliosis and neuroinflammation. And we'll hear a lot about neuroinflammation today, and other poorly defined damage. And this other poorly defined damage is probably going to tell us a lot about what we think the mechanisms are of the results we found in our study today. So there are some hypothesized mechanisms of damage. So like I said before, fractionated whole brain irradiation can induce deficits without detectable anatomic changes. And that led to the idea that maybe it was neurogenesis and cell proliferation within the brain. There are two distinct niches within the brain, and one of those is the subgranular zone, the dentate gyrus of the hippocampus, that is important for neurogenesis. And the hippocampus is very important for consolidation of long-term memory, in addition to other types of cognitive functions. And so the development of hippocampal sparing radiotherapy techniques was designed to save this cell population. Now here's one of the first studies showing these things, showing the decrease in new neurons and immature neurons following whole brain irradiation. And what you see here are doses in 2 gray up to 10 gray. So these are like single fraction doses here with this 2 gray. And this is the number of Ki-67 or positive cells, so cells that are proliferating within the subgranular zone of the hippocampus. And you see a decline at all of these doses to about 48 hours post-radiation. And this is DCX, a marker of new neurons. And what we see here is a decrease at all of these doses. And over time, 2 months post-radiation, we see a decline in the number of new neurons with dose, but an increase in the number of oligodendrocytes. So this suggests that the microenvironment of the brain is changing following radiation. And even though cell proliferation and decreases in neurogenesis are important, there are other things going on within the brain. And so adding to that, more recently, neuroinflammation is now viewed as a key mechanism in normal tissue injury following radiation. We have reactive gliosis and active microglia that occur early on and continue for months after radiation therapy. We have increased pro-inflammatory cytokine release, infiltration of peripheral immune cells, and this seems to be a very important mechanism of this neuroinflammation. And we also have microvascular damage that further alters the progenitor environment and leads to cognitive deficits. So this is the hypothesized timeline of damage, and we see many of these early changes that lead to changes in dendritic structure. These are very important. So this is different than a mechanism of simply decreasing neurogenesis and cell proliferation. Here, we're actually changing how neurons look and possibly how they function. So adult neurons, and that's really important. That's been a shift in what we view the mechanisms of these injuries are. And this idea that different forms of this early damage persist, and we don't exactly know how this early damage is related to the long-term damage. But I'm going to show you today that neuroinflammation could be a key mediator of that. And so we have a decline in these neuronal and supporting cell populations, leading to loss of cognitive function. And while most of this is focused on the hippocampus, there has been a recent recognition that hippocampus alone might be too simple of a concept for understanding radiation-induced cognitive dysfunction. So as we move from whole brain irradiation to stereotaxic radiosurgery, SRS has been used for treating brain tumors because it spares more healthy tissue than whole brain irradiation. However, studies of patient cognitive function after SRS are limited, and the ones that are out there report opposing effects on quality of life, probably because they're measured at different times in the survival of patients. This quality of life is especially important for this group because treatment of the primary cancer is improving and life expectancy is increasing. While the prognosis can still be poor for some folks, their post-treatment life can be made better by increasing their quality of life. And so we need to understand how SRS can do that. And there are limited preclinical models studying the long-term effects of SRS. And so that's where we've tried to fit in our study here. And so preclinical studies of normal tissue injury are typically focused on non-tumor bearing subjects. And this provides a great method to study the effects of radiation on normal brain tissue. It misses an important aspect of the potential of the effects of tumor breakdown, damaging the brain tissue as well. And so most preclinical studies are examining either single doses such as 10 gray or fractionated whole brain irradiation. I highlighted a few studies here, especially the one by Parahar from Charlie Lamoli's lab, looking at stereotaxic radiosurgery with a total dose of 10 gray. We're going to go back to that because we found some similar things with our higher dose SRS. So there's some interesting comparisons there to be made. And some additional models. There are very few models looking at tumor-bearing animals that survive long enough to be able to assess cognitive function. So we think this is an important thing to do. That's what we tried to do in our study today. So in patients, like I said, radiation is delivered to the tumor and the normal tissue. And so we really want to understand with our model how these cognitive changes might be addressed in animals and preclinical models that survive tumor eradication. So some of our questions were, are long-term outcomes similar in a preclinical model that has a tumor? And does degradation of the tumor impact the response of the normal brain tissue? And so our research questions here were to develop a tumor-bearing model in which we could study the long-term effects of radiotherapy, specifically SRS, on cognitive function. And our initial hypothesis was that SRS would eradicate the tumor and would cause limited cognitive dysfunction. From the few studies that are out there, the effects on cognition were more transient than whole brain irradiation, even fractionated whole brain irradiation. And so our additional questions was, would SRS have an effect on neurobehavioral function? So just SRS alone, and would tumor eradication affect any of the neurobehavioral outcomes that we were assessing? And so this is our study timeline. And so we trained C57 black 6 mice, these were all male mice, on 2 different behavioral tests prior to any manipulation to have a similar baseline of behavioral performance between all animals. And so one of those tests was the rodent psychomotor vigilance test. This is a sustained attention test where the mice have to monitor a light within an operant chamber and respond to that light whenever they see it come on. They're then rewarded with food. And so that light can take anywhere from 3 to 10 seconds to come on. And so they just have to monitor that. It's a very simple reaction time test. We also assess the social odor recognition memory test, which is done in the home cages of animals. And I'll explain that in more detail in the next few slides. So following baseline training of our animals, once all animals were meeting the same performance criteria, the animals were split into different groups. And some of those animals had tumors implanted, and these are 9L rat glioblastoma tumors, and some were exposed to sham surgery. So within the tumor implantation group, we had a no treatment, so a tumor plus no irradiation group, and then a tumor group plus stereotactic radiosurgery at 40 gray. So this will be the tumor plus SRS group. We also had a sham group, like I said, and this group received sham surgery, but no tumor implantation. And they also received 40 gray of SRS. So these are the sham plus SRS groups. The no treatment group was just followed with bioluminescence imaging of the tumor and MRI. And all these animals succumbed to their tumor within 3 weeks of implantation. The animals that received radiation exposure were then tested in our behavioral paradigms for up to 40 weeks following radiation. They also received multiple bioluminescence imaging and MRI imaging at these various time points. So we're going to have time points at 2 weeks, 10 weeks, and at 79 weeks. And then at each of these time points, 2, 10, and 79, we also have immunohistochemical staining and aerochrome staining for myelin. So I want to provide you some detail on the social odor recognition memory test, because this is what I'm going to be talking about the most. This is where we found some of our deficits. So this is a test, a 2-phase test where rodents are asked to identify their own odors and that of a novel odor. So we have familiar or self odors, and these are wooden beads that are placed into their cages to acquire their scents. And then from a conspecific, they receive the odor of another animal, and of a second animal. So on the first phase, which is called habituation, they receive 3 of their own odors and 1 of a novel odor, a conspecific they've never experienced before. And typically what you see here is illustrated by these bars. They spend little time exploring their own odors, and they spend the majority of their time exploring this novel odor, because rodents can tell the difference between their self and conspecific odors. For this mouse test specifically, the recognition test came an hour later, where mice were then asked to remember the previously experienced novel 1 odor, and then they received a brand new odor from another conspecific that they had never experienced before into their cage. And what we expect for an animal that is unimpaired is that they spend little time examining their own odors. They should spend less time examining the novel 1 odor because it was experienced on the habituation phase an hour earlier. And they should spend the majority of their time exploring this novel 2 odor because that is the newest odor to them, and rats are attracted to novelty. So we infer from this behavior that they're remembering the experience of the novel 1 odor and that they have intact recognition memory. So impairment here would look something like this, where they spend equal amounts of time exploring the novel 1 and the novel 2 odor. Our stereotactic radiosurgery paradigm delivered 40 gray to the tumor site, and the tumor was implanted within the striatum. And you can see this dotted line is going to detail for you in a cartoon form the area of the brain that was exposed to radiation. And you can see here from the sagittal and the coronal views. So we delivered 40 gray to the tumor center and then had decreasing dose to the areas around it. So the green is about 10% of that 40 gray. And you can see the 80% line is this orange line pretty close within the tumor area. And so importantly, what we're exposing here is a portion of the cortex, which is here, the corpus callosum, which is the white matter here, the striatum, which is where the tumor was implanted, and this portion of the lateral ventricle. So those are all very important regions to the brain. However, we purposefully avoided exposing any of the hippocampus on either side of the brain to radiation exposure. And this was done with the idea that the hippocampus could be important for either one of our behaviors. And so we avoided any exposure to that brain region. This is to demonstrate tumor growth without SRS. So in that group that I mentioned to you that received tumor implantation but did not receive any radiation exposure, you can see here that the tumors grew large. The tumor was tagged with M. cherry, so we could follow the growth by fluorescence microscopy. You can see here that there was microglia at the border of the normal tissue and the tumor, and little microglia infiltration into the tumor when there was no treatment with SRS. So we delivered a single dose of 40 gray at this time point here, which we delineated as week 0. So this was 9 days after tumor implantation, which gave the tumor time to grow within the mouse brain. And you see here we have equivalent patterns of growth from bioluminescence between from -8, which was day after tumor implantation, to -1, which is the day before radiation exposure. And you can see that similar growth within the quantification of the BLI signal here on the right panel. Once we delivered the 40 gray of SRS, we followed these animals again. And what you'll see is that to the animals that did not receive radiation exposure, the tumor continued to grow. And within 3 weeks, all animals had succumbed to the tumor. However, on the SRS group here, what we see is the tumor begins to shrink. And by week 4, we see no more bioluminescence signal from this tumor. What's important is that we still have a signal at week 2, and you can see this quantification here, and that's going to become important in our behavioral data when I show you that. So just remember that we still had tumor in the brain at week 2 following radiation. And so 40 gray treatment with SRS did prolong the survival of tumor-bearing mice. So it completely eradicated the tumor by 4 weeks following radiation. And that was what you saw in the last slide with the bioluminescence signal. And here you see increased survival of the tumor plus SRS group. So there was no difference in survival rates between tumor and SRS and the sham animals who also received SRS. So throughout this study, which lasted a year and a half post radiation exposure, we did not lose an animal in the sham plus RS group or the tumor plus RS group. So this gave us a nice period in which we could study neurobehavioral function of these animals. This is to show you the immunohistochemistry of the tumor disintegration following this single dose of SRS. And what you see on the right, the left panels, I'm sorry, in A, C, and E is IBA1 staining, and this is a marker of activated microglia. The tumor again is in red. It fluoresces with M cherry, and DAPI is cell nuclei. And so at week 2, following radiation exposure, we see infiltration of activated microglia into the tumor, and the tumor is disintegrating, but we still have tumor evident. At 10 weeks following exposure, the tumor is gone, but we still see activated microglia, and the same thing at week 79. So this is approximately 18 months following radiation exposure. For GFAP, which is in astrocytes, we will see increased GFAP, but we see limited GFAP in the tumor core. But at week 10 and week 79, we see a greater signal of GFAP, and we'll talk about that in the next few slides. So by week 10, by week 4, with the bioluminescence, the tumor was completely eradicated, and that was also confirmed with the IHC data we show here. And so these are, I'm going to talk to you the next few slides about the behavioral data that we found. So we tested again the mice in the social odor recognition memory test at baseline, which you're seeing here. And on the left-hand side is the tumor plus SRS group. So this is baseline before any tumor implantation and any irradiation. And this is the sham SRS group on the right-hand side. And so what you're seeing here is that all groups had equivalent baseline performance. And so you're seeing only the novel 1 and the novel 2 data just for clarity. So what we typically expect, like we described before, was an increase in exploration of the novel 2 odor, because it's the newest odor the animals are investigating compared to the novel 1 odor. And that's what we see here at baseline. Importantly, that performance was maintained at 2 weeks following radiation exposure. And within the tumor group, there was still tumor evident at this time point. So that's important. So we don't think the presence of the tumor is having anything to do with the behavioral deficits that we're going to see in the following weeks. So we then tested the animals within this paradigm every 4 weeks up until 22 weeks following exposure. And so at 6 weeks, we start to see a differentiation between the groups. The tumor plus SRS group now are starting to show a behavioral deficit, a memory deficit here, with no significant difference between the exploration of the novel 1 and the novel 2 beat. However, the sham plus SRS group are maintaining their baseline-like performance. At 10 weeks, however, both groups are showing behavioral deficit here in the social odor test. And at 14 weeks, both groups maintain that deficit. However, what we see is that in the sham SRS group at 18 weeks, they again are maintaining baseline-like levels of performance. However, this is appears to be a chronic and persistent deficit in the tumor plus SRS group. So again, they're showing no difference between novel 1 and novel 2. Out to 22 weeks, that deficit is maintained within the tumor plus SRS group. However, that deficit is now completely gone in the sham plus SRS group. So what's important here is that the tumor plus irradiation group is showing a persistent deficit in social odor memory, whereas the sham plus SRS group is showing a very transient deficit in this type of memory. We also looked at the psychomotor vigilance test, which again is that sustained attention test. The data at BL is the baseline, so before tumor implantation and irradiation, and you can see that both groups maintained equal levels of performance. The triangles are correct responses, and the circles are lapses in attention. And so what we see in both groups is a similar type of performances, where immediately after radiation exposure, within a few weeks, they're showing a transient decline in correct responses and a transient increase in attention lapses. So more errors and a slower performance in the attention test. However, that transient deficit is gone in the following weeks, and both groups were able to maintain baseline levels of performance throughout the 40-week testing period. So what we have is very specific deficits on memory in the tumor plus SRS group, and very transient deficits on attention in both groups, and a transient memory deficit in the sham plus SRS group. So to try to understand the mechanisms of these deficits, we next went in to look at inflammation within the regions around the tumor and where the tumor was disintegrating, to determine if there was neuroinflammation within these regions, and if that could explain the differences that we were seeing. And so here on the left, on the top, is GFAP staining at week 2 following radiation, and IBA1 staining on the bottom. And you see tumor plus SRS and the sham plus SRS. And the insets here, we're controlling we're showing you the ipsilateral and versus the contralateral side to the SRS. So what we see at 2 weeks is an increase in IBA1 staining in the tumor plus SRS group. And you can see that in the quantification here on the right, and the blue symbols here. So this is significantly different from both the sham plus SRS and just sham animals levels of IBA1 staining. We also have increased levels of GFAP staining in the tumor plus SRS group. While we do have increases in inflammation in the sham plus SRS group, we have an exacerbated level of inflammation in the tumor plus SRS group. And so we are attributing this increase in inflammation to the disintegration of the tumor. So at this point, we still had tumor evident, but we had microglia infiltration in that tumor region, and we believe that this is a response to the breakdown of that tumor. At week 10, these are the same targets, so GFAP and IBA1. However, what you see here is that we still have increased IBA1 and GFAP staining in both groups compared to sham. We don't have that exacerbated level of IBA1 staining in the tumor plus SRS group like we did previously at week 2. And this final quantification is comparing weeks 2 and 10 for GFAP, and this is the white bar is sham plus SRS, and we can see a slight increase in GFAP staining from week 2 that has declined by week 10, and a similar pattern in the tumor plus SRS group, but we had, again, a greater level of GFAP staining in the tumor plus SRS group. Interestingly, in the sham plus SRS group, while we had an increase in IBA1 staining relative to sham controls, which you can see above, there was no difference between week 2 and week 10 IBA staining in this group. Whereas we had an exacerbated level of inflammation in the tumor plus SRS group at 2 weeks post-exposure that had declined to the levels of all other groups by week 10. We next looked for demyelination or neuronal loss throughout these regions, where we were also looking at neuroinflammation. And at the same levels at 2 weeks and at week 10 following exposure, we did not see any changes in irochrome staining, which is a marker of myelination. And so we saw no demyelination within any group in the quantification you can see here. We also did not see any neuronal loss, and we used NU-N, a marker of adult neurons here in green. And you can see the quantification of sham versus the tumor SRS. So we saw no decrease in the total number of neurons and no demyelination. We next looked into the hippocampus to determine if there was any inflammation in the hippocampus. Now remember, we specifically spared the hippocampus in delivery of our SRS treatment. And so we looked, however, within the hippocampus to see if we found any increased levels of neuroinflammation. And what again you see here is week 2 with GFAP and IBA1 staining. And we also see at week 10 as well. So we have the contralateral and the ipsilateral hippocampus. And with the quantification here, you can see that we found no differences from sham animals to sham plus SRS and tumor plus SRS. So we did not have an increased level of neuroinflammation within the hippocampus. So we only had exacerbated neuroinflammation within the brain regions surrounding where the tumor was eradicated. We also looked for edema, necrosis, or breakdown of the blood-brain barrier in the hippocampal region. And so these are MR images at week 2 and week 10. And we have no abnormal signs with on the T2 weighted images at the top, indicating edema or necrosis. And we have no enhancement of the gadolinium contrast in the bottom panel, showing no breakdown of the blood-brain barrier within the hippocampal region. Following these relatively acute, up to 10 weeks, we looked at the end of this study at approximately 18 months, we looked at our markers of neuroinflammation. So again, GFAP and IBA1, our markers of adult neurons, and our markers of demyelination. And what we found in the tumor plus SRS group was that we had continuing inflammation around the tumor region. So in that cortex region of the brain, and you can see that quantification here from the images, and the blue dots here are the tumor plus SRS group. So like an elevated signal of IBA1 and GFAP staining, but again, no loss of neurons. And so this signal from the new N staining was similar between sham and the tumor SRS group. We also found no evidence of demyelination by the erythromycin staining again. So in the quantification here, similar outcomes between the sham and the tumor SRS groups. We also looked at MR images, and these MR images were taken in animals at 6 weeks, 10 weeks, 24, and 79 weeks. And so these animals, went back into the behavioral assays. And so it was really nice to be able to track these measures on the MR images and then be able to look back at the behavior and see how these animals progressed. And so again, what you see on the top is the T2 weighted images, and then the images on the bottom with the contrast. We did not see up to a year and a half post radiation exposure any loss of integrity of the blood-brain barrier. However, what we did see was an increase in brain atrophy, specifically in the ipsilateral hemisphere of the region that was irradiated. So this is all tumor plus SRS groups here. And so at week 6 and week 10, we saw no brain atrophy. And these are the images here. The arrow is indicating the tumor region. But at weeks 24 and week 79 post-radiation exposure, we saw a significant decrease in, brain atrophy at week 24 and week 79. And while there's a slight decrease again at week 79, these two groups are not significantly different from one another. So essentially equivalent atrophy that started at weeks 24 and persisted to week 79. And this was in the ipsilateral hemisphere here with where the tumor was. So in summary, SRS with 40 grays eradicated the tumor, and we had no recurrence up to 18 months, which was the end of our study. Tumor degradation appears to result in persistent neuroinflammation, memory deficits, and brain atrophy in the irradiated area. The tumor did not cause the deficits. So again, remember, we still had tumor present in our animals at 2 weeks post-radiation, but we did not see a deficit on the memory test at 2 weeks post-exposure. And that transient deficit we saw was in both groups, the tumor plus SRS group and the sham SRS group. So it's very unlikely that the presence of the tumor in the brain caused any of the behavioral deficits that we're seeing. However, we believe that tumor degradation, so after the radiation exposure, was associated with elevated inflammation, and that tumor degradation was what was leading to these persistent neurobehavioral deficits. We also found neuroinflammation that was present for up to 18 months within the regions surrounding the tumor. And the sham plus SRS group, so animals that received that 40 gray dose, had only transient deficits and limited neuroinflammation. And so we think this is very supportive of the fact that SRS spares brain tissue and doesn't cause as much damage as fractionated whole brain irradiation. We found no hippocampal inflammation. That shouldn't be surprising because we didn't irradiate the hippocampus, but nothing that within the cortex made it to the hippocampus in terms of inflammation, any type of bystander effect, at least from the measures that we took. We did not have a loss of neurons anywhere we tested within the hippocampus or within the region surrounding the tumor area, and we had no loss of blood-brain barrier integrity throughout the duration of the study. So we believe from these results that the tumor breakdown is an important factor to examine in our preclinical models. And like I said before, most of our preclinical models of radiation-induced brain injury do not include the tumor, and they only include normal tissue effects. But the tumor appears to be, degradation of the tumor appears to be a really important thing in the health of the brain following radiation exposure. And so just to compare our results to a few papers that are within the literature, we looked for other papers using preclinical, primarily rodent models of SRS, and we were able to find the Parahar study. And what's nice is within this study, they assessed behavioral outcomes at 6 weeks post-exposure. And what's interesting is we also had a 6-week time point, and in our SRS only group, we showed no deficit at 6 weeks. And the same thing with the Parahar study. Now their dose was much lower at 10 gray. But they found no deficits at that 6-week time point when they were irradiating one specific region of the brain like we did with our SRS. However, they were comparing their results to whole brain irradiation, and they found that SRS resulted in much fewer deficits than whole brain irradiation did. The importance, though, of avoiding the hippocampus is unclear for us. So we avoided the hippocampus and still resulted in long-term memory deficits in the animals that had a tumor implanted and that tumor degradation. In the Parahar study, they had multiple groups where they irradiated and avoided the hippocampus with their SRS, and the deficits were different depending on what behavioral tests they were looking at. So some that were explicitly hippocampal dependent showed a deficit. So the importance of avoiding the hippocampus is unclear at this point, I think, because we've shown deficits in behavioral tests that aren't explicitly dependent on the hippocampus when irradiating other portions of the brain. And we believe that tumor degradation, like I said before, can exacerbate neuroinflammation and then damage these regions around the tumor that are important for neurobehavioral performance. So I want to make it clear that just because we didn't irradiate the hippocampus doesn't mean that the fact that another brain region was damaged that communicates with the hippocampus. Now you might get a cognitive deficit because the communication between those regions has been changed, and that's probably something that's happening within our study here. Some of the limitations of our study that we didn't look at peripheral immune cell infiltration, and we know that's an important part of neuroinflammation within the brain following radiation exposure. And so in future studies, it would be interesting to see if peripheral immune cell infiltration is different in the tumor model versus the animals that are only receiving radiation alone. We also didn't assess any cytokines or vascular factors within the brain regions of our animals, and those would have been important mechanistic things to learn. So we're hoping future studies can look at those in addition to cell proliferation. So while we looked at total neurons with our marker, we didn't look at Ki67, for example, which is a marker of cell proliferation. And it would have been interesting to look at that marker within the lateral ventricle, especially on the ipsilateral side, because that ventricle received a good portion of our dose. And so it would have been interesting to see if there were differences within Ki67 between the contralateral and ipsilateral side. The Parahar study that I noted here looked at Ki67 and BRDU staining and DCX staining within the contralateral and ipsilateral hippocampus. And they found a really interesting compensation in the contralateral hippocampus for a loss of cell proliferation within the ipsilateral hippocampus. So I think it's something that needs to be investigated in the future, especially in a model that has tumor degradation as a factor. So I think we can also use this current model to investigate hypofractionation of SRS, which could lead to maybe less inflammation based on tumor degradation. So maybe if we eradicate the tumor a little bit slower, we might be able to save some of that inflammation. And again, that's a hypothesis that we would need to test. So what, in these last few minutes, I want to sort of hypothesize about what the mechanisms could be. So with recent information coming out for radiation-induced brain injury, there seems to be an appreciation now that adult neurons can be damaged by radiation exposure. And it's not, that neurons are dying, it's that this very intricate communication that they have that's detailed here can be damaged. And not only damaged by direct hit from radiation, but also from a continual environment that is exposed to neuroinflammation and release of cytokines and things like that. And so what a lot of studies have found recently is a decrease in dendritic spines and altered protein expression at the synapse, an altered number of synapses, and altered and changes in the receptors that are expressed. And so this can all have an effect on cognition, because obviously appropriate cellular communication is necessary for these behaviors. So we don't just have to damage or eliminate cell proliferation to see a deficit on cognitive function. And so one of the regions that's important for many of these behaviors is the prefrontal cortex, specifically the medial prefrontal cortex, which is shown here in the rat, and it's very similar in the mouse, and which is color-coded here to match those regions within the human brain. And so I primarily study rats. And so we asked the question previously, what is the role of this region in the behavioral test that I showed you? So in the social odor recognition memory test. And what we found, by using a viral vector to silence the medial prefrontal cortex, we were able to induce a deficit like we saw with radiation exposure. So again, no preference for that N2 odor, so similar exploration of those two odors. So by inhibiting neuronal activity within the medial prefrontal cortex, we were able to induce a deficit in the encoding and consolidation of those recognition memories, but not in the recollection of those memories. So it's possible that radiation exposure can damage neuronal communication within this region, and that could then lead to these deficits that we're seeing. Some interesting studies with radiation exposure more recently have shown in preclinical models that radiation exposure disrupts the neuronal activity within the prefrontal cortex. And I think our data here with the viral vector is in support of this in sham animals. Radiation can also disrupt the hippocampal to PFC communication. So these regions are in communication with each other for all of these different cognitive domains, especially the ones that we've shown you in the talk today. And that these changes happen even though there are no change in adult neuron numbers like we saw today. And these changes have been attributed to things like loss of dendritic spines and altered excitatory inhibitory signaling, probably from glutamatergic synapses, because treatment of these animals with memantine can reduce some of these deficits. And memantine has been used in radiotherapy patients as well, and it's showing some promise as a way to eliminate some of these deficits. So it's more than just cell proliferation and loss of those cells within the hippocampus. We have other important brain regions that are involved in cognition and that can be damaged by radiation. And like I mentioned, one last region that is important is this subventricular zone. It is the second neurogenic niche within the brain. And we did not look in our study at cell proliferation there, but this is I just showing you again here on a brain slice, sort of the tumor or where we delivered radiation in our study. And now while our sham SRS group showed very limited cognitive deficits, it's possible that eradication of the tumor resulted in some change to neurogenesis within this region. We have other studies showing that no changes in neurogenesis in this region, and we still have deficits in social odor memory tests in rats with different irradiation exposure. So it's not clear that this region would be involved, but I think it's something important that we would need to look at as well, given that it's another neurogenic niche. However, I want to echo the sentiments here at the bottom from a recent review saying that worrying only about the hippocampus might be too simplistic of a view of radiation-induced cognitive function, and we really need to take a whole brain approach and to understand the radiosensitivity of these different brain regions and how we might best deliver exposure while eradicating tumors and stopping their recurrence. And so some final thoughts here on this study. So SRS in our study can eliminate brain tumors, which it did. And we chose the 40 gray study from previous work where 20 and 25 gray were unable to eliminate that 9L gliosarcoma. So 40 gray was successful and the animals survived as long as sham animals. But the breakdown of the tumor within the animal could be a concern and could exacerbate inflammation. However, we think there's potential for therapies that inhibit neuroinflammation to reduce these deficits resulting from SRS tumor ratification. And I've just highlighted a few here. There's many other studies using different types of neuroinflammation inhibitors. But I highlighted this one here because Feng et al. In the eLife paper is actually using a mouse model of glioblastoma. And while the animals, while they increased survival, they didn't completely eradicate the tumor. They were able to use PLX 5622 to prolong survival of these animals so that they could actually test some neurobehavioral performance, so primarily novel object recognition. It's a really interesting study. They used whole brain irradiation, whereas we used SRS. So I think there is potential, though, for compounds like these within our model to determine how we can modulate neuroinflammation to produce better cognitive outcomes. It's possible that SRS fractionation could be used. There was a recent paper talking about flash fractionation. So flash is this ultra high dose rate radiation, and flash fractionation appears to be as effective as flash. So that could be possible for SRS as well. And our study also supports the fact that SRS is less detrimental to normal brain tissue than whole BI. Than whole brain irradiation in preclinical models. And that's comparing that to that normal tissue model with Parahar. So I thank you. And I just want to acknowledge all of my collaborators at Hopkins. So again, all this work was completed at Hopkins. I was formerly in the Division of Behavioral Biology with Bob Hines, and then all the folks from radiology and radiological science, and then from radiation oncology and our funding sources. So I'm going to close my slides so I can see everyone's faces.
Thank you very much, Katie. That was a
You're
really
welcome.
exciting project, and it shows us how important it is to choose the right animal model. And not many people actually have studied these kind of things in a tumor model, so that's really important. If anyone has a question, I would say just unmute yourself and ask away.
Sure.
So that was a nice talk. I really enjoyed that.
Thank you.
So it's hard. I know you showed evidence and you argued pretty strongly against direct effects of the tumor, but it's sort of hard to see how that this big gaping hole in the it's hard to see how that couldn't affect something. I mean, so that's my first kind of question, and I'm kind of wondering if there's any way to, I mean, it would make the experiment more complicated, but if you could put like some gel or something, just some tumor-sized chunk in there to kind of, that's presumably inert, to maybe get around some of that. Because that's my first wondering, but my real question is, given the loss of neurons much later, do you think, much post-radiation, do you think that could be due to like transecting axons and things with the radiation and having a sort of delayed death, like preventing neurotrophic factors and stuff like that from happening?
Yeah, that's a really good question. And, it's unfortunate that we didn't look at something at some of those neurotrophic factors. So it's very possible that the fact that we're substantially changing the microenvironment within the brain, which I think is what we're doing. And I think having the tumor there is changing it even more severely than in a normal tissue model where there is no tumor. So it's very possible that with the tumor there and the degradation and the hole that we've created, that we're changing the trophic factors that are being released, or they're not being released, or that, they just can't repair the damage as well. I'm not sure exactly where to start with those mechanisms. I mean, clearly neuroinflammation is ongoing, and we know that. And there's plenty of data showing that if you eliminate that, you can prevent these issues. But there's clearly more happening to the brain. There's more damage at the axonal level and the synaptic level. And I think there's potentially a lot of targets there that could be used. So I wish I had a better answer. But, I think it's something to think about, because we just don't know, exactly the integrity of every single axon, right, within the brain at that point.
Good. Any other questions? So I am still fascinated and not still trying to understand the neuroinflammation. Say it's clearly going on, but why would that neuroinflammation be there up to 18 months if your tumor is gone such a long time ago?
Yeah,
for a large part.
I'm not sure I have an answer for that. Clearly something has changed within the brain. And I think what we're missing is important. We're missing important factors. So we don't know what any cytokines are being released within the parenchyma. So we didn't analyze those. We don't know levels of cell proliferation. So I think we have a lot of missing pieces here, unfortunately. We have the suggestion that chronic neuroinflammation is bad, but after we get lower into those mechanisms, we don't have those answers. We could have lost spines, we could have lost synapses, which is probably, we could have altered protein expression within the synaptic structure so that the neurons can't communicate as well as they could before. There's many targets, I think, that we would have to look at this point. It's unfortunate that we didn't do a lot of those things, but we can only do so much, I guess. Yeah.
May I ask a question?
Yeah, sure.
Yeah, that's a wonderful talk. Yeah, thank you.
Thank you.
I'm quite interested in the blood-brain barrier. So I just wondering that if you know more about the mechanism that after irradiation, how that destruction with the blood-brain barrier, and also do you think that the destruction of that would contribute to the brain damage or the behavior change?
Yeah, I mean, it's possible. So from what I understand, I'm not an expert in the brain barrier, but from what I understand is that activation of the endothelial cells are going to cause the blood-brain barrier to allow infiltration of peripheral immune cells. And so we don't know in our study, for example, how big of a component that actually was to the injury that we were seeing. So we didn't find any, gross changes in the blood-brain barrier, but that doesn't mean that those factors, say VEGF and CCR2 and all those different factors weren't altered in some way that is causing increased infiltration of immune cells and maybe micro damage that we couldn't find within our MR images or something. Yeah, or other types of microvascular changes that we missed because they weren't necessarily things that we were looking for. Yeah. It's a great question, though.
Thank you. And then Dr. Barger has his hand raised.
Thanks. Yeah, I'm very sympathetic and supportive of your arguments that lots of regions contribute to cognition and even particularly memory outside the hippocampus. But I wonder if your tumor was injected into the striatum, why you didn't test motor function more specifically. And one thing that's really intriguing to me is growing evidence that the striatum participates in really complex motor patterns like obsessive compulsive disorder, which aren't always thought of entirely as motor. But I wonder, do you have some tests of repetitive behaviors or something like that that could be applied to mice in this kind of model?
Yeah, so the attention test that we used, the animals, while it's not like a rotorod or it's not doing gross motor behavior, they do have to move their bodies to respond to a light, and we track their reaction times within millisecond ranges. And so we found no changes within a millisecond range of their reaction time, just those transient changes I showed you. So for us, we think that's evidence of no motor impairment. However, I understand that those aren't, I'm not putting them on a asking them to walk a long distance to see, you know, how their coordination of their motor ability is. So it's possible that there are some motor impairments that we weren't able to find, but I don't think those are going to produce the deficits that we have. The social odor test is not, it doesn't require necessarily motor movement to be able to perform it. And all the animals were able to move to the front of the cage within the time allotted and explore the beads. We did see, on the other hand, we would have loved to have done some behavioral testing in the animals that had the tumor, that did not. We had them in the lab and we tracked them following implantation of the tumor. What we saw, though, as that tumor started to expand within that brain region, these animals clearly were impaired. You know, loss of writing, clear impairment in their motor abilities, which we never saw. I mean, gross motor changes that we never saw in the animals that had the tumor plus SRS. So I think we would really need to be careful about the test that we design to potentially get after that. Because we had a nice comparison with the animals that had the tumors growing, and they had clear gross motor impairments that precluded us from doing any studying of their neurobehavioral function, because we couldn't separate out what was a motor impairment versus what was a memory impairment at that point.
Thank you. Very good question.
Yes.
Does anyone else have a question? Before we. Great. So with that, I'd like to thank Katie one more time for this excellent talk. It's very nice to see you too, Katie.
Thank you for the invitation. This was very fun. It was a very fun study to share and to do. So I appreciate you all listening.
Yes. Thanks everyone for attending, and have a very good rest of your Monday.
You too. Thank you, guys.
Bye.